Okay, so what do I want to talk about now? I want to talk about their general ecology, and I want to talk about the word of the day. If we think in general about bryophyte biology, we have to unthink almost everything we know about vascular plants. And most of us are more familiar with vascular plants, and because we can grow them in gardens and we sort of have a feeling for what they need and what their requirements are and in everything about them. And in a short form, bryophyte biology just differs from tracheophyte biology in pretty much every way. And so it's not good to think about the bryophytes as being lower plants. That tends to make biologists mad, feel denigrated, because the bryophytes are not lower. They're really successful. They're one of these groups that have increased their diversity over time, but they just took a different approach than the vascular plants did. The vascular plants definitely got bigger and they got more complex in their sporophyte, but the bryophytes got more complex in their gametophyte and came up with solutions or retained solutions that were just different ways to live on land. So we'll talk about not all of them, but we'll talk about some of the most important ones. So each of them have their advantages and disadvantages, just like any other kind of organism. So it's better not to think of the organisms that are around today, better not to think of any of them being lower or ancestral relative to any others. They're all survivors. They've all been around. Every organism alive today, its ancestors have made it to the present. So they've been successful in that sense. So here's just a summary of some of that huge list of ways that bryophytes differ in their ecology and evolution from vascular plants. And I'm not going to talk about all of them, but I'm going to talk about most of them. We've already talked extensively about the haploid dominance, that the bryophytes all elaborated their gametophytes much more than they did their sporophyte. Somebody mentioned in chat plasticity. All plants are pretty plastic. Plasticity means that you can achieve different morphologies with the same genotype. Plants in general are more plastic than animals are. So you get with vascular plants or with bryophytes, you can get a wholly different kind of looking leaf in the sun and in the shade. And it's not because their genotype, their genes are different. It's because of the direct effect of the environment. So all plants do it, but bryophytes seem to have really a lock on it. Bryophytes are so plastic. It's one of the things that drives one crazy when you're trying to identify them, is you have to learn the variants that many of our fairly easy to identify species, once you know them, come in a lot of different forms so that you don't necessarily even recognize them in the field. But when you look at them closely, you go, okay, yeah, that's what it is. So those of you who know bryophytes know about ceratodon, which is kind of our freeway shoulder weed of the bryophytes. It grows on roofs, and it looks really different. It's very often something you confuse. You collect it and wonder what that is, and then you realize, oh, it's just ceratodon. Okay, and then here's our word of the day, Poikilohydry. We're going to go into that in detail. Goes along with desiccation-tolerance. Bryophytes are in that in a big way. They reproduce in a different way than the tracheophytes do, or at least than sea plants do. Ferns and bryophytes share the need to have actual water in the environment for their gamete to swim in, whereas sea plants don't need to have that because they've internalized the gametophyte. And I'll explain more what I mean about superorganism. But given their small size and the clumpy way that some of them grow, the actual unit that's acting as the individual is really the clump and not the stem. So in some sense, they're kind of like a beehive, in that bees face the world not as individual bees, but as a hive of cooperating animals. We have a whole lot of asexual reproduction. Somebody mentioned that in the chat too. Bryophytes go in for various ways of reproducing asexually, just forgetting about the sexual reproduction entirely. So we're going to that. The mere fact that they're small actually gives them different rules. If you are into such things, if you're into physics, you know that the boundary layer effects, so temperature and movement of air and water that are close to surfaces, is really different than out in the flow. So if you're looking at the flow of liquid through a pipe, your average flow of the liquid through the pipe is different than what you would see if you look right close to the surface of the, the inside of the pipe, the wall of the pipe. You can have turbulence there and not have turbulence in the main flow. So we have to think about life in a different way when we're down in the boundary layer. Some people have used that as a title. So bryophytes live in the boundary layer, whereas angiosperms don't. They're so small, they're living in the physical space, which gives them a really different temperature and water relationships than the big plant that's sitting right next to them. So we'll talk about that a little bit. Then related to all of this stuff, and I'll go into this in more detail, but it looks like very often the main challenge for the bryophyte is the physical environment, trying to grow anywhere, and not quite as much competition with other bryophytes or other organisms, but particularly with other bryophytes. So I'll go into why that might be. The combination of the haploidy with maybe lower amounts of genetic variation, and for whatever reason, genetic morphological change tends to be really slow in bryophytes. So as we go and look back at the fossil record, way back to even before there were angiosperms at all, there are many moss families and genera that look the same as they do now. So they're kind of survivors, and they do their evolving perhaps largely on the physiological side where you can't see it. That's on the functional side, and not necessarily in the morphological side of things. Okay, so here's the word for today: Poikilohydry. So Poikilohydry is from the Greek root poikilo, refers to changeable. Another word that you probably do know is poikilothermy. So poikilothermy would be a word we would apply to the cold-blooded so-called animals, like a lizard or a snake, where the temperature of the animal is roughly the temperature of the environment that it's in. It doesn't have a mechanism to put its temperature higher than the environment it's in. Whereas, so that would be poikilothermy. Poikilohydry means changeable water amount. And that would be that your water content as the plant equilibrates to that of the surrounding environment. So to put it really simply, it would be like a piece of tissue paper on a rock. If it rains, it gets wet. And if the sun comes out, it gets dry. It doesn't have a mechanism to keep the water content high like the vascular plants do. No roots. Internal conduction is not a big thing in them, and they dry really quickly. So that's going to influence pretty much every aspect of their biology. So if there's one word you get out of today, it should be poikilohydry. That's our watchword, and we'll talk about that, and maybe in discussion we can talk about more, but it just affects everything. Going along with poikilohydry, if you're going to be poikilohydric, you better also be desiccation tolerant. Desiccation tolerant means that you can handle having your tissues get completely dry and then come back to life again, which none of the mammals can do, no reptiles can do. There are just one or two insects that can do it. It's very rare in animals. With any kind of animal, once they're completely desiccated, they're just dead. They have no mechanism to come back. Most vascular plants are the same way. Most vascular plants, even cacti, are not able to come back from being completely dry. So if you go away on vacation, you forget to water your cactus for a long time, you come back and they're all dried up. It doesn't matter how much water you put on them, they're dead. They won't come back from it. So a cactus has a different strategy for dealing with water. It holds onto water and keeps its water content higher than the environment, like a human does. We have mechanisms, we drink water, and we have ways of trying to hold the water inside of us so that we don't immediately dry out. So we're going to talk a lot about desiccation tolerance. These two things kind of go hand in hand. They're not absolutely linked, but if you're going to dry and wet frequently, you've got to be able to handle being dry. Poikilohydry, this is a beautiful little moss that some of you may have seen in the coast redwood forest, particularly in the northwest part of the state. This is Herbaria, and is a mainly tropical group that has a few representatives up here in the temperate flora. I don't know if anybody's ever seen these. They're beautiful, and you can even see their individual cells. They're hugely delicate. I just put this in to be evocative of the idea that given that these bryophytes only have one cell thick leaves, they're really going to be quite intimately connected to their environment. They're going to lose water out into the environment when it starts to dry, and they're going to take water directly in once it gets wet. That has to do with not just the physiology of desiccation tolerance. It's going to have to do with cellular materials from the plant going out into the environment and vice versa. So we'll talk about that a little bit more, but it has to do with their pollution sensitivity, with their relationship with other organisms that live around them, and so forth. So if you're going to think bryophyte biology, you got to forget about roots and bark and cuticles and all that, and think about a structure that is much more intimately connected with the environment. It's got a lot of chemical communication going on, which I want to talk more about. So here is what desiccation tolerance looks like. Stacy showed a couple of different Centrichia pictures in her introduction. This is one of the Centrichias. This is Centrichia princeps, the prince of mosses, which is one of the common ones you see in the coast ranges. It's the most common Centrichia. So I think you guys have seen it. Here it is, I just tore the clump in half and poured some water on half of it from my canteen. So these are exactly the same plant, but this is what it looks like dry, and this is what it looks like wet. It can look like this dry side for decades, maybe even a century, and it can still come back to life. And so that's a super interesting phenomenon. That was made by KQED, although we did help them do it, but their photographers are fantastic. They're able to get images we never would be able to get. But the point of that little 3-minute segment was that because of the Poikilohydry, because they're taking in and releasing water right over the cell surface, it supports a little community of tiny organisms, and the rotifers are one of the kinds of organisms. I don't know if anybody's ever seen a tardigrade. They look like a little Michelin man kind of a thing. Tardigrades are really cool. Water bears, they're called. They're also desiccation tolerant animals that dry up and rehydrate just like the plants do. There's a bunch of algae and fungi, all of which dry up and reconstitute. So when the moss is getting wet, it isn't just the moss, it's also its community of associated organisms that's coming back to life. And so it's a very different sort of situation. You have a whole community of organisms that can go into suspended animation and then come back to life. And you can't say it's a less successful strategy than big metazoa like us or big angiosperms that control their environment more and keep from getting dried, but it's just a different sort of a strategy. In the video, they mentioned this message in a bottle kind of a thing. They wrote themselves a message, if you caught that. When a moss is perfectly happy and hydrated, it's been wet for a long time, it builds up its reserves. It also stores away a signal. Just a quick review of a little bit more genetics. We have our genome, our diploid genome, and the way the genome gets expressed in proteins is that each gene gets transcribed into what's called RNA. You guys have probably heard of that before, if nothing else, because COVID is an RNA virus. So RNA is like DNA pretty much, but just has a few differences. And organisms lay a template against their genome, against their DNA, and they replicate, and they make just a copy, like we would do a Xerox copy of a piece of paper. They make a copy of the little segments of the genome in RNA. Then the RNA goes off, what's called the messenger RNA, goes off to the ribosomes. And at the ribosome, which is the protein synthesis factory, the message lays down in a certain place, and then a protein is assembled. So you have transcription of DNA into RNA, and then what we call translation of RNA into proteins. So that's how all organisms, from bacteria through animals and plants, that's how everything makes proteins. The normal way, when there's a stress of some sort, the normal way that an organism responds to that is by turning on certain genes and making messenger RNA and making a protein that it needs to deal with high temperature or something like that, or parasites, or virus attack. The mosses do a slightly different thing. They are making messenger RNA, but they make it ahead of time in preparation. They don't need it at the moment, but they're making it ahead of time. They package it away in a stable way during the dry phase here. So while the plant is dry for a long time, months, years, it already has this messenger RNA packaged away inside of itself, so that when water is added again, the first thing it starts to do is make proteins from the stored message that was already present in the before times. And then it will make what it needs to repair itself, and will repair its photosynthetic machinery, and then begin to photosynthesize again. And then eventually it gets back to this happy hydrated state. So it can stay in this state for quite a while. It can stay in this state for quite a while. The rehydration phase takes hours, a few hours. It's very quick as compared to a few of the resurrection vascular plants. If you did take Carl's teratology course, you probably saw that some of the lycophytes and some of the ferns are desiccation tolerant. Common ones that we all know are the polypody that grows on tree trunks, and the golden back fern that grows on soil banks around here. Those also, and also many of the selaginellas, those can recover from being completely dry, but they take days. Their mechanism is slightly different. They take days to do it, whereas the moss is doing the recovery in a matter of minutes and hours. It can take as little as 15 minutes to get back to normal. And the difference really between those two strategies, the one that a polypody uses and the one that a moss uses, has to do with, if you have a little bit more warning, you can do things to protect yourself right before you dry so you don't get as damaged. Whereas if it's a catastrophic thing that the drying just happens really quick and you can't prepare for it, then you got to be ready ahead of time. So the analogy I always use to explain this to people is, at least us Californians are used to earthquake preparedness. We can never tell when an earthquake is going to happen. So each of us should have a stored water and blankets and flashlights and the things that you would need after an earthquake emergency. That's kind of what's going on here, is that the moss is storing things that it's going to need in the future, but it doesn't need right now, rather than waiting until the earthquake happens to try to fix itself up. Whereas the Selaginellas and the desiccation tolerant ferns have more warning. They know a few days ahead of time that water's running out because they're not poikilohydric, they're endohydric, they're bringing water up inside. So they have time to do a few things that to protect themselves. So they have more of a protection-based strategy, whereas the bryophytes have more of a recovery strategy. They go ahead and get damaged and they can just repair themselves. It's the best they can do in their circumstance. Okay, so where does Poikilohydry appear on this phylogeny? Here's that same phylogenetic topology again with our three big groups of bryophytes here. And then this whole group is the vascular plants, the tracheophytes, tracheid-bearing plants or vascular plants. Seed plants are one of the groups. And then here's that fern and equisetum lineage that we talked about. And then diverging earlier is the lycophyte lineage, which has Selaginella, Isoetes, and Lycopodium. And there are scattered, pretty much all the bryophytes are desiccation tolerant. Then there are scattered members, but not all members within Selaginella. So some lycophytes, some but not all ferns, and a few angiosperms. There's not a single gymnosperm, which is desiccation tolerant. And there's a few angiosperms, but there's none in our flora. So there's none that you might know very intimately. It's a very unusual condition for an angiosperm. There's a lot of problems with it. So the one place that there's desiccation tolerant in all of these guys is in the seed. So once we get going in the genomics, we now have sequenced genomes of all of these, and we have sequenced genomes of a representative set of the land plants, of the tracheophytes. It appears that the genes that were involved in vegetative desiccation tolerance originally in the ancestors and their descendants here, the bryophyte lineages, once the plant itself became not desiccation tolerant, and this is something I'll ask you about, there's a teaser for the next discussion, is why didn't all plants stay desiccation tolerant? We know for sure their ancestors were, and probably the ancestors of humans were too. So why didn't we keep the ability that we used to have in the past to do this? It would be kind of handy if, for example, a corn plant could dry up completely and then come back when you add water. Why not? That would be it seems like a advantageous thing for all plants. But plants in the vegetative tissues mostly gave it up here. Then it was re-evolved a few times in a few of the groups of land plants, but a major place it occurs is in the seed. If you remember seed biology, once an ovule gets fertilized and becomes a seed, many of them are actually desiccation tolerant. So they can be dried out and they don't germinate or come back to life until water is added again. So there's a whole bunch of plants that have seeds that are desiccation tolerant, even though their vegetative body is not desiccation tolerant. It's a different kind of desiccation tolerance in a seed, because with a seed, once you break dormancy, it's a one-way street. Once you break dormancy, you got to go ahead and germinate. So if you're germinating some seeds on your windowsill, once they start growing, don't let them dry out again, because they can't go back to being desiccated. It's once they break dormancy, they have no way to go backward. Whereas the original condition found in the bryophytes is a reversible sort of condition where they can go wet and then dry and then wet and then dry and then wet and then dry. So that's a little picture of what we think happened. And what I want to talk about in the discussion period is kind of what were the driving forces of this. So here's this social organism bit, and even involves the community ecology stuff we were talking about with the rotifers and the associated animals and algae, which is that if the plant is losing water or it loses its cell content when it dries, and then it pulls back in materials when it's wet, that's like a pump that's putting stuff out into the environment and or bringing things back into the cell. So it, in many cases, is feeding these organisms the basis of the food chain that's in the moss clump is the sugars that are being lost by the plant. One of the damages that happens to a plant when it dries is to have the membranes, shrink down and break, and they actually lose stuff out into the environment. They don't lose their cellular integrity, so they have little sugar molecules going out into the environment. So many of these associated organisms are picking up on that. And then, which affects that, it also affects communication. There's good evidence that the plants around a given stem of a moss are responding to chemical communications from other nearby mosses. And when you have a hormone that acts across individuals, that's what we call a pheromone. So there are good indications of that there's communication going on. And then the reason that they're so susceptible to pollution would be because they're bringing the environment right back inside of them. So if there's some kind of pollutant in the water that they're in, or in the air shed that they're in, they will accumulate in the tissues of the plant these pollutants much more quickly than a trichophyte will, because a trichophyte's a little bit buffered, because you have to get water into the plant. It's got to come from the soil, up through the root hairs, up through the roots, and then up through the vascular tissue. So Poikilohydry has lots of effects on pretty much on everything, how they relate to their environment, how they relate to other organisms, how do they relate to each other, and so forth. So Poikilohydry, that's why it's the word for the day. It's really important. So how do they deal with water? They're 1 cell thick. This is a transmission electron micrograph of a cross-section of Centrichia, the moss. And you can see it has a surface open to the environment on one side and open to the environment on the other side. So the water is running right in and out of there. It turns out that what mosses and liverworts mostly do, instead of internal conduction, is external conduction. They have mechanisms for bringing water up from the substrate. We saw it happening in that video, actually, and opening up their leaves, bringing it up their leaves and bringing it out the surface of their leaves and bringing it right on out to the active part of the leaf from below. Many of them do this like Centrichia does by having open dead cells that function to bring water up from one leaf to the next across the sheathing leaf bases, and supported by the rhizoids here, which we'll talk about. Rhizoids are not roots, but they're they're more like root hairs. They're little 1 cell thick thread that's coming out from the stem. And then the cell surface itself, when you look closely, has these really interesting papillae on it that look like little antlers. Looks like a little Stonehenge here. That's a highly magnified view up here of what the cell surface looks like. You can see the papillae in cross section here. So papilla, very common in mosses and also in liverworts, is an extension of the cell up into the environment. And what that basically is doing is acting like a little snorkel. Water fills up the channels in between the papillae when the plant is fully hydrated and happy. The ending, the little tip of the papilla, actually sticks out above the water film and has a way, you can see the chloroplast, this is the photosynthetic organelle that's in the cell, is sticking up into the papilla. So it's getting the CO2 that it needs to do photosynthesis, and it's letting out oxygen sort of through the papillae. So they are actually pretty good at moving water around and even storing water. They just mostly do it in external structures instead of internal structures like trichophytes do. They need this water for reproduction. Now here we're talking about sexual reproduction. They need the free water, which is something that ferns and lycophytes need also, but the seed plants don't need. So to look closely at what an archegonium looks like, we've introduced archegonium before. That's the female gametangium. There's only one egg cell in each. And then there are cells that are eventually going to dissolve away to make a opening, and this will open at the top. And the amazing sperm, this is a drawing reconstruction by Karen Renzaglia. The sperm cells are amazingly complex. They're asymmetric. They have two flagellae, but they're not inserted in any kind of a symmetrical way. And they have a very complex and characteristic structures for each taxon. So this very complex sperm is produced in the antheridia, which is here. This is an antheridium. Each of those little tiny squares in there will eventually become a gamete, a male gamete. Those male gametes have to swim and find the opening to the archegonium and swim down and fuse with the egg. So it's not easy to do, and it requires a continuous film of water. The mini mosses have some kind of a form of a splash cup or something that looks like a flower, but it's not really a flower, but it's a place where if a raindrop hits, it can bounce water out of it that contains some of the sperm. But their gamete dispersal distance is very short, a matter of inches. So they're not going to be doing like a seed plant can, like a pine tree that can release its pollen into the wind, and the pollen could go miles. You're not going to get these swimming miles. These are lucky if they can swim, an inch before they run out of energy. So sexual reproduction is kind of difficult. Maybe we can talk about this in discussion, why they have this mechanism. It's kind of a clumsy mechanism, actually, and it means that especially in drier places, they have a hard time actually doing it. They have a hard time of actually mixing. So that brings in asexual reproduction. It's so hard to do sexual reproduction that many bryophytes, have some sort of asexual propagule, and some have pretty much given up on sexuality. We have many mosses that we don't even know that they produce either male or female gametangia at all. There are some that appear to be just clonal. Here's something I haven't mentioned so far. Thinking back to basic botany again, your moss, all of the bryophyte lineages, the 3 bryophyte lineages, plus the lycophytes, plus the ferns, so all the same land plants that have this free-living gametophyte also have a form of cell division where only a single apical cell divides to produce all the other cells that are going to come. So that's called an apical cell meristem, where there's a single cell. In the seed plants, you have a bunch of cells leading the way, each of them leaving descendants that make the plant body. It's much more complex. They have a multicellular meristem, many leading cells. So you can get sectors within a leaf, for example, and you guys have seen variegated leaves. You can have a sector where some parts of the leaf have one genotype and others have a different. And what we call it somatic mutation. So somatic just means body. So somatic mutation is something that happens in the body of an organism, which in the case of organisms that grow from an apical cell, if there's a mutation in division of the apical cell, it's going to be passed on. So that you do get a lot of genetic variation, even though we said that because of haploidy, you might expect less, you get a lot still, because over the millennia, as bryophytes grow and as they do their mitosis, they make mistakes. So even in purely clonal species, even the ones that have no known sexual reproduction, you get plenty of genetic variation, as it turns out. So we'll come back to that in discussion too. That's kind of interesting. So we got all kinds of asexual propagules. This is Alicomnium androgynum. This is also a redwood forest thing. Many of you have probably seen it. It's on stumps. in nice moist forests. And it looks like it has a sporophyte, right? It looks like a sporophyte sticking up there, but it's kind of like not organized into a capsule. It looks like a pom-pom, like a cheerleader might have. What's happening there is each of those are gemmy, and each of those can grow into a new little plant, but there's no sex has been involved. That's just the same genotype as the mom. So that's one of many, many ways that they would reproduce asexually. Orthodicranum, this is one that we see in on logs in our forests in California. You might have seen this. It has these long, skinny leaves, but here's a finger of a person. If you rub against it, you come away with your finger covered with leaf tips. So this particular one reproduces asexually by breaking off the leaf tip. And then if you were to put your finger down on the substrate, or if a little chipmunk had done it instead, the moss can grow in that place. So this has a different type of asexual propagule. Liverworts do it too. Here's Calopogia, which is very common leafy liverwort. We'll talk about the organization of liverworts later. But for now, I just want to point out these pom-poms that it has at the end of the stem. Those are asexual propagules as well, each of them being able to grow into a new plant. In my Centrichias that I love, Centrichia papilosa and Centrichia pagorum grow on the same tree. For some reason, these kind of like urban trees. So if you go to most of the parks, like the parks in Berkeley or the parks around in Northern California, look closely at the trees and you're likely to find both of these. They're little Centrichias, you can tell because they have a hair point on each of the leaf. But this one produces gemmy on the upper leaf surface, so Centrichia papilosa. Whereas Centrichia pagorum, its gemmy are quite different in developmental origin. They are little tiny leaves. So they're miniaturized leaves, whereas these are outgrowths of leaves. But both of these are quite successful in spreading because each of these will grow into another little plant. And I put in a blender here to remind me to tell you that when we propagate mosses in the lab, we just grind them up. You can grind them up. I learned that technique from a bonsai grower. Bonsai artists have a beautiful arrangement at the bottom of their tree where they have mosses representing elements of a landscape. So you have mosses representing a valley, and you have, with like a grassland, and other mosses representing a forest. They're long time ago realized this fact that you can grind them up in a blender, and you can kind of paint them on where you want them, and then they'll grow into the plant that you want. So I don't know if anybody ever saw my Mythbusters episode. I've been on TV one time in the Mythbusters where they did an experiment to see whether moss grows on the north side of trees or on a rolling stone or on any kind of stone. And the technique for grinding up the moss to put it onto a rolling stone and a not rolling stone was just a blender with a little bit of buttermilk, which is a technique learned from the bonsai growers to kind of make it sticky and hold together, and then just painting it onto the surface. We can talk about that. If anybody saw that, it's kind of fun. You can still find it on reruns. So to finish up talking about moss and bryophyte ecology in general, I just want to talk about basically where they grow. We have this stereotype view that they like to grow in places like redwood forest, where they certainly do like to grow, very lush, wet kind of places. But if we want to think that they only grow in those kind of places, we would be really wrong and doing them an injustice. So they love to grow in the coastal rainforest habitats, and they grow huge there. But they like to grow in lots of other habitats. They live in basically every terrestrial biome. There are none that are truly marine. Unfortunately, I keep traveling around the world and looking in different oceans and never have seen them. And so they appear not to be in any real intertidal anywhere, although there's a few species that are in the very highest tide level. There's two different ones that we see in the Pacific. On the Pacific coast, you can see not where they're ever covered by the ocean, but where they would get some ocean spray kind of way up in the above the tidal zone. Quite characteristic to those places, but they live in all kinds of other habitats other than the ocean, including fresh water. They still like to grow in water. So they grow on pretty much everything, and the kind of substrate they grow on is usually quite specific for a particular moss or liverwort. They don't grow just on any rock, but they're going to grow on a rock with a pH of a certain amount. They're going to grow on some trees as hosts and not other trees. And despite the preconceptions, I've made bets with people at various times when we're on CNPS field trips. In any one location, you can often find more bryophytes than you can vascular plants. So it's a myth that bryophytes are somehow less successful than the vascular plants, because every vascular plant, you might have 6 trees in a particular place, 6 kinds of trees, but each of those kinds of trees has 50 kinds of bryophytes living on it. So since the bryophytes use the trachyphytes as substrate, you can often have a lot of local diversity of bryophytes. One reason why many people have gotten into studying bryophytes, many people who got their chops in botany with vascular plants will have a we'll do in an off-season kind of way, we'll do bryology, because you can see them any time of year. You don't have to wait for the spring, and you can observe them right while you're observing your vascular plants that you want to observe. Here's a typical North Coast forest here, and in a habitat like this, the number of bryophytes would rival the number of vascular plants in a given, let's say 10 meters square. This is a really famous moss here that if we ever get around to proposing one as a state moss in California, it'll be this one. This is Dendroalcea, which unfurls as it hydrates, sticks out like a little fir tree. I'm pretty sure you guys have all seen these in the field. It's a very prominent thing when you're walking in one of the Coast Range forests. So you get many, many different kinds of bryophytes that like to grow on trees. Here's one growing on a rock. You get many other bryophytes that like to grow on rocks, and they're usually distinct. So if you are a rock growing bryophyte, you're usually not going to be on a tree. Then there's a whole bunch of soil dwelling bryophytes. I just picked a liverwort, Rixia, here for an example. And again, if the rains have been enough this year, you can find these right now. They're ephemerals, little tiny dichotomizing. I saw some in Tilden Park. So look closely at the soil banks when you're walking. You just might find some of these ephemeral mosses and liverworts kind of right now. But see, we'll talk about liverworts next, but they're quite, this is quite obviously a liverwort. It's got the a thallus organization, where here's a moss. And then this I put in just to remind me that there are many bryophytes that still grow in water. So even though they're land plants, they live in fresh water. This is the fountain moss, Fontanalis anaerotica. That is quite common in our coast ranges in a really clean water. They are really sensitive indicators of water quality because of this Poikilohydry thing I was talking about, that many species of aquatic bryophytes hate any kind of water pollution. If the nitrates are too high in the water, it'll disappear. But if you find Fontanalis growing in a stream, you're pretty sure that you've got a nicely clean stream. We actually had a whole workshop on soil biocrusts a few years ago, and I saw somebody in the chat said they were there. So the biocrust community is about the opposite of those freshwater aquatic communities, I guess you could say, where they're living in very dry situations, dry land, desiccated situations. This is just west of Las Vegas in Nevada, where we worked. This is our research group here. We've worked on one of the centricheas there, but they form these amazing communities seen in close up here, which have not just mosses. Here's centrichia, but it also has lichens, different kinds of lichens. There's a yellow one there and a white one there and a green one there. It also has free living blue-green bacteria and also green algae. So this would have many animals in it, like rotifers and the water bears and little tiny mites and spiders. And this is a whole community. And this is one of those instant on communities that can sit there all summer in the sun, but boy, you add water and it comes back to life. And it's like the living skin of the earth. The crypto crust, the biocrust, would be much more common on our landscape if they weren't as disturbed. Off-road riding or cattle grazing or even hiking trails destroy the biocrust, and then you get a very different soil situation in the desert where it can erode because the particles are free. When there's an intact crust, as you see here in the lower left, there is no free soil. The soil is not actually exposed at all to the air, and you can see a little tracheophyte getting going here. The desert tracheophytes like to, in fact, in some cases are required to germinate and get established within this stable soil crust community. And they won't get started in a place like right by a sidewalk here where everything's been broken up and there's no biotic soil crust right here where we're standing, probably because we're standing there. But if you went a little bit away from the viewpoint here, you would find nice, well-established biocrust. So that's an interest of mine, and it's just indicates that bryophytes live in really counterintuitive places, like out in the middle of the desert. Our SOBI-free, those of you who are going on SOBI-free in 2 weeks in the Mojave, we're going to be able to show you lots and lots of biocrust. And then, of course, they do live in more mesic places. These are centricheas, but there are many other mosses that like to grow on soil in woodlands, or this is in a chaparral kind of an area, or way up in the alpine. This is up above tree line. Bryophytes tend to go way up higher than trees do. And in Arctic and Antarctic situations, when you get down to the Antarctic Peninsula, there's only a couple of asker plants that can even live there. And there are many, many lichens and liverworts and mosses. So Antarctica is one continent where the bryophytes are actually completely dominant. Even when you get into the high Arctic, you find landscapes that are dominated by bryophytes. So they kind of come into their own as compared to tracheophytes in really high altitudes or high latitudes. So coming back briefly to this small stature business, any organism that's small, just by virtue of being small, is going to be experiencing the environment differently than a big organism. So in some ways, mosses have more in common in aspects of their biology to salamanders than they do to a bird or a mammal. And I have a colleague that works on salamanders, and the biogeographic patterns and things of salamanders are actually closer to bryophytes than they are to bigger land animals, because they're just small. They're only influenced by their immediate environment. They're in the boundary layer, which means that they're so close to the surface that the temperature and wind speed and water that they're experiencing pressed to the bark here is actually different in kind than what they'd be getting if they were an inch high. And so we do find that over time, this, just like salamanders, you often find bryophytes lingering on in a place because of some past climate that is gone totally. So maybe, California and many aspects of many parts of the US were tropical at one point. They're not now, except for the southern tip of Florida. So we have still bryophytes from like that Herbaria that I showed you whose ancestors are basically tropical, and they're still just hanging around in these micro environments, as long as they can find a microenvironment to live in. This one you probably have seen, it's common around here. This is going to be Frulania, which is a very kind of burgundy colored oppressed liverwort. Here's Polytrichum, which is quite common. And this is actually one of the leafy liverworts we'll look at later, called Bassania. But as I said before, many bryophytes have trouble getting established. They have trouble getting their gametes to fuse. And then once the spores get dispersed, the spore has to find a suitable place and get established, which is a tough thing to do, a tough life when you're a little tiny spore and you can be abraded away or lost in many ways. So in any but the most hydric kind of situations, so I'm not talking about bogs here, but most places, the bryophytes don't actually fill all the habitat that's potentially available to them. This kind of an environment right here is a very stressful one, but bryophytes love it. This is where you're in near freshwater streams and you're covered sometimes and you're not covered other times. That's a very prominent bryophyte habitat, but they do have trouble getting established and remaining because of water flow problems and things. So it turns out that paradoxically, unlike tracheophytes, very often when you have one bryophyte present, it's actually beneficial or conducive to other bryophytes being present. So this gets back to this idea that natural selection in the drier communities would be more the organism versus the environment and not the organism versus other organisms.